I hope you know that to study practicals, you need to have a firm foundation in theory. If your theory is not firm, you always get a problem in practicals, because practicals is a summary of all theory topics you have studied. So you need to, when you're studying your theory, you need to have a firm foundation, so that when you come to this type of practicals, you are able to apply your principles of theory, because theory is the basis for the practicals. Today I want to talk about a simple concept. I'm not very certain of how much theory you've covered. So I want to inquire from you, have you studied simple principles in the theory of like osmosis and diffusion, active transport, which are found in the movement of materials across the cell membrane? Have you studied osmosis?
Yes.
If you have studied osmosis, that's the concept I want us to apply here. And I think you can define osmosis. Who can define simply, simple terms, the process termed as osmosis? Any person who can define osmosis? Hello? Who can define osmosis? Now, if someone put up the hand. Nobody can define to us osmosis? Hello? Yeah?
Teacher, I'm going to try. I'm going to give the definition.
Uh-huh.
I'm going to give the definition we used to use. Osmosis.
Hello? Continue.
Teacher.
Mm-hmm.
Osmosis is the movement of water molecules from their region of high concentration to their region of low concentration across a semi-permeable membrane.
Yes, it's a movement of water molecules from where they are highly concentrated to where they are of low concentration across a semi-permeable membrane. And if you check that definition, if osmosis, water is moving from where it's highly concentrated to where it is of low concentration across a semi-permeable membrane. Someone can even use another term, or if you ever heard of diffusion, our diffusion is the movement of molecules, but osmosis is the movement of water molecules. But diffusion is the movement of molecules, whether solute or water molecules. So long as any molecule is moving from its high concentration to where it is of low concentration, then we could say there's diffusion of molecules. But osmosis slightly differs from diffusion because it has a condition that whatever is moving must be water molecules only. But diffusion is so long as there is any movement of molecules, whether solutes or water. But osmosis is only water molecules. That's one condition. Another condition for osmosis is that movement of water only without crossing a barrier, which in this case, we are saying a semi-permeable membrane. If water is not crossing that barrier, then we will not refer to it as osmosis. It will remain diffusion. So with osmosis, we have to have a barrier. And in this case, the barrier we are taking to be a semi-permeable membrane. Why do we refer to it as semi-permeable? Is that it is selective in permeability. It only allows water molecules to pass. And it stops solute molecules, so that we don't have any other molecules diffusing. And then you have only water diffusing. Now with that, if we compare osmosis and diffusion, you could find that osmosis is a subset of diffusion, because diffusion is broader since it considers both solute and the solvent, in this case water. But osmosis only considers water molecules moving. So I can as well define osmosis as the movement of, or instead of using the movement, I can define it as is the diffusion of water molecules across a semi-permeable membrane. Automatically, when I talk of diffusion, from the definition of diffusion, whenever there is diffusion, there must be a concentration gradient. And when I talk of concentration gradient, I hope you don't get lost. Gradient is the difference. And when I talk of concentration gradient, I mean the movement should be along the gradient, moving from its high concentration to its low concentration. So I can as well define osmosis as the movement, the diffusion of water molecules across a semi-permeable membrane. I hope you can see now the difference between the two. For diffusion, any molecules diffuse doesn't require semi-permeability or a barrier, so long as there's movement, but that movement should be along a gradient, should be moving from high concentration to a low concentration. But osmosis is movement of water molecules from its high concentration to low concentration. Now, when I talk of from its high to low, then I can even substitute those many words by using diffusion. I can say the diffusion of water molecules across a semi-permeable membrane. Someone who has a firm background in theory will understand that when I say it's diffusion of water molecules, I'm referring water moving from where it is highly concentrated or where it is too much to where it's of low concentration. Now that process for us in the practicals, we may not ask you to define osmosis, but what we really need from you is the ability to apply that process. And applying that process, you have to base on the changes that occur to living tissues. Because that process cannot be seen occurring, but it depends on the effect of that osmosis, so that you can tell whether there have been osmosis or not. And for that reason, I want us to relate osmosis to living tissues. And in that case, I want to use plant tissues. And when I talk of plant tissues, someone may ask me, where is a semi-permeable membrane? I hope you know from your senior 1 or even senior 5, you're going to talk of a plant cell. A plant cell, structurally, it has 2 membranes or boundaries. There is what you call a cell membrane, which is common to each and every cell. Each and every cell should be bound by a membrane, which you call a semi-permeable membrane, which you call a cell membrane. Another term you would have used, you call plasmalemma. You find the same thing. But then, a plant cell has an additional wall. An additional wall which is outer to a membrane, cell membrane. That additional wall, you all know it's called a cell wall. And the 2, cell wall and cell membrane, they define structure and chemical composition. When I talk of structure, I mean physical, and then chemical composition, the molecular structure. A cell wall, as you know, its main composition, the cellulose, it is fully permeable because it has numerous large pores, that cell wall. Though it is rigid, not elastic, like a cell membrane, but a cell wall is rigid, but with large pores. Being with large pores, that means it is permeable to all molecules, whether solutes, which are always large, all water molecules. They can all pass through freely across the wall. But then, inner to the cell wall, we have got a membrane, which you call a cell membrane. Structurally, it differs from a cell wall because it's thin and elastic. But a cell wall is not elastic. A membrane is thin and elastic. A wall is thick and inelastic, structurally. But any chemical, a cell wall, we said, is basically fundamentally made up of cellulose, a network of cellulose, the fibers made up of cellulose. If you remember, to make cellulose, we use beta glucose, that one you must be knowing. But then a cell membrane is made up of chemically phospholipids, which are not, if you know, permeable to water. It's not permeable to polar molecules like ions, but it is permeable to organic. Like if I add a solvent of lipids, it can easily pass through because they are of the same nature. Now you find that since it is composed of phospholipids, it stops the fusion of so many molecules, major which are polar, which are charged. So it has some pores in it. Those pores are tiny. And if you remember, those pores are made up of protein. They are called protein channels. Those protein channels, they are channels crossing the membrane. And those channels have pores which are liquidated, they have some water molecules. And those pores can allow water molecules to pass. Some are also special to allow polarized solutes to pass. But then if I go back to our practical, the basis here, if I go in details of the membrane, let's take the membrane being semi-permeable. Why? Its chemical nature, it allows only smaller molecules to pass. But back in our mind, we know that even solutes can pass because of those different protein channels. You know, some protein channels can even pump molecules across with the use of energy. That's called active transport. When you come to coordination, maybe if you are doing some private reading, you find that there's what you call potassium sodium pumps, which can pump those two across, in and out. But then there are those even have which do facilitated diffusion. They make molecules to diffuse without using any energy, which we call facilitated diffusion. It's also done by protein channels. So that's the nature of the membrane chemically. So you find that it can very nicely allow osmosis because it can stop solutes to pass and then allow water molecules to pass. But then application of that process, I want to use plant tissues. But before I use the plant tissues, we need to know the relations. Because to have different concentrations, one to be highly concentrated compared to the other, what we do is to dissolve solutes in water. When you dissolve solutes in water, then you can change its concentration. There are more solutes from your chemistry, you may call it molality. For last year, I may not so much go in details of molality, but it's all about molality. There are more solutes that are dissolved in a solution. So long as I don't change the volume of water, if I have two solutions, A and B, I have solution A, and I have another solution B of same quantity. Now, if I put here, let's say 1 gram of salt, can be table salt or in a sodium. And then I put 2 grams of salt. Hope you all testify here. This one will be concentrated. It will be of dilute concentration. Low concentration. From your chemistry, I hope you know that symbol means concentration. And this one will be of high concentration. Why? A has little solutes dissolved, and then B has more solutes dissolved. So technically, if I don't use this low concentration, because this one high concentration, low concentration is a descriptive kind of term. I'm just describing the solution. Now this one is of low concentration. The other one is of high concentration. But there are some terminologies we can use, many of them, which could be applied here, mean the same thing. Now, if I use a term, let's say, if I use a term like a hypotonic solution, and then hypertonic solution. If I may ask, between the two, A and B, which one can be referred to as hypertonic solution? Someone to answer that. Your network. Yeah. Oh. Yeah? B and A, which one is hypertonic? Yeah?
A is hypotonic and B is hypertonic.
Good. This one is hypotonic. Hypotonic solution. And this one is the hypertonic. I think since you're all scientists, when I talk of hyper, someone is hyper, high. And then hypo means low. So when I say hypotonic solution, I'm referring to concentration. This is hypotonic, this is hypertonic. When I talk of hypertonic solution, I mean it contains more solutes than the other. Automatically, if it is one solution, I can apply the same one, but it's more meaningful if there are two, because that word is comparative. B can be hypertonic to A, but you may find that if you have a third solution, a third solution can be, if it contains more solutes, then it can be hypertonic to B. So these terms are relative. It is comparative compared to the other. This is hypertonic. Why? It contains few solute molecules. This is hypotonic. Why? It contains, this is hypertonic, sorry. Why? It contains many solute molecules. This is hypotonic because it contains fewer compared to B. Now, I want also to use another term. Here. Now, between A and B, remember, there's another term I want us to apply here. Okay, if I use the term pressure, osmotic pressure. Between A and B, if I have high osmotic pressure and then low osmotic pressure. Here, I can use H of P. I'm abbreviating here. And here I'm going to use law of P. But these are not convention. I'm just applying here. Now, I think you ever heard of osmotic pressure. Before I talk about it, between A and B, which one has a low osmotic pressure? Someone to tell us. Solution A and B, which one do you think would be considered as having a low osmotic pressure. Hello? Are you there? If you're not certain, you could say, please, no, I'm not very sure, then we'll go about it. I can throw some light about it. Osmotic pressure, which one would be having?
We're not certain.
You're not certain.
We're not certain. I think that's why we're silent.
Okay. Osmotic pressure. Okay, what I want to tell you, if you're not certain, same time, just say, we are not certain, and then we talk about it without taking too much time. Osmotic pressure is just referring to concentration. So when I say osmotic pressure, I'm referring to concentration. It's directly proportional to amount of solute dissolved. The higher the solute dissolved in the solution, the higher the osmotic pressure. So long as I've maintained the volume of the solvent constant. Like here, if I have a glass of water having 500 mils, that is mils, I mean centimeter cubed, would be A. And I have another B having the same volume. If I put here, like I did here, I put 1 gram of, let's say, solute. And then I put 2 grams of the solute. When I talk of osmotic pressure, it is basically referring to how much solute dissolved or concentration. The more solute dissolved, the higher the concentration, and then the higher the osmotic pressure. The less solute dissolved, the lower the concentration, and then automatically the lower the osmotic pressure. But what's the effect of osmotic pressure? Osmotic pressure, why should we consider it? Now, if this is solution A, and then I drop, let me enlarge this one. Okay, large. If I have solution A, this is a beaker. I'm making it larger because I want to fit in a cell, a plant cell. And this one, this is A, and this one is B. All have 500 cc. And then I drop here a plant cell. Let me say this is a cell wall. I'm talking about the application of this term. Now this one has, let's say these plant cells have the same concentration. I've got the plant cells from the same plant. A plant having this, okay, having cells from the same plant, that means that those cells have the same concentration. We don't know the concentration of the plant. But then we are going to assume if this one is hypotonic, like you say here, and then this one, we say the A is hypotonic. This is A, which is hypotonic, which is of low concentration. That means that if I compare now this solution A, which I've taken to be hypotonic, and then I compare to this plant cell, so long as I say that this solution A is hypotonic, I can even, since you know the ground is here, if this is hypotonic, and this one is hypertonic. What does that mean? When I say B is hypertonic, then you have to ask me, hypertonic to what? Like if I say you are taller, it's different from someone saying you are tall. You are tall is not comparative. You're not comparing with anything. That means that maybe by standards of the nation or that society, that particular society where you are, someone say that person is tall. At the back, is using the standard of that society. But then when I say you are taller, then you have to ask, taller to who? What you consider to be short to someone could be taller to the other. So here when I say hypotonic, then you ask hypotonic too. Now, to what? Now since I put here plant cells, A and B, when I say this is hypotonic, that means that this solution, where I dissolved 1 gram of sodium, it could be sodium chloride, anything soluble. That means this plant cell here, if the solution is hypotonic to the cell, that means the cell is hypertonic to the solution outside. Likewise, here when I say the solution is hypertonic to the cell, that means the cell is hypotonic to the solution. So now, since you have two solutions here, for example, in A, the surrounding solution is hypotonic to the cell. That means the cell is hypertonic. Then you go back to the definition we started with, where we say osmosis is the diffusion of water molecules across a semi-permeable membrane. Technically, that diffusion is okay. I told you when I say that diffusion, I mean the movement of water, because osmosis is only water moving, of water molecules from where it is highly concentrated to where it's of low concentration. Now, if this solution here is hypotonic, the cell is hypertonic, then there is a gradient. There's a difference in concentration. There's a solution enclosed in a plant cell. That solution, we can call it a cell sap, but I can call it maybe a cytoplasm. Now, we have created a gradient The surrounding solution is hypotonic. The cell sap is hypertonic. If I use the cell sap, I mean the liquid part of the cell. Then osmosis can take place. I've created one condition, a difference in the concentration. But then we say there must be also another condition, which is semi-permeable membrane. This plant cell, what you see here is a cell wall. I've just drawn a cell wall, like what I've drawn there, if I do like that. It is a cell, this is a cell wall. I don't see a cell membrane. Here, what I'm drawing is I'm drawing what I see if I put a plant tissue on a microscope. Forget your theoretical plant cell where you use the one we have in texts. They use what we call electron microscope, which has a very high resolution power. What I mean, it can magnify to a greater magnitude. I hope you compared a light and electron microscope. What appears as one in a light microscope, in electron, it will appear separate. That means it can magnify, it has a higher power magnification, which I've called resolution power. So here, this is a wall. So someone can ask me, where is the membrane? The membrane is fully closed, attached to the wall, and then to be seen as one wall. But there is a membrane. And I've said, a wall is permeable to all solute and solvent because it has larger pores. But then what it brings in osmosis is the membrane, which is semi-permeable. The solids tend to diffuse, but then will be stopped by a membrane. But then water will diffuse. So here osmosis can take place. If I transfer this one here, outside here is hypotonic, and then inside is hyper. So I've removed it from the beaker, but I put here the arrow. I hope you understand that. For clarity. Now I may ask you, this cell, based on our definition, someone should be ready to tell us. Will this cell A, based on that definition, will it lose water or gain water? In what direction will water move? Will water move into the cell based on the definition, or will move outside the cell? Who can tell us? What will be the direction of water movement? Any A?
It will move inside the
cell. Yes. Water will move inside. Why? Outside, it is hypotonic. Inside is hypertonic. And we said water moves from where it is abundant, high concentration, to where it's low. But then you may wonder, even here inside there is water. Why can't water get out? It's just on that principle. The principle is it moves from low, from highly concentrated, where it is highly concentrated, to where it's of low concentration. Automatically, if I'm to use the term concentrated and or low concentration and high concentration, that term, concentration, I'm referring to the solute. When I say low concentration and high concentrated solution, I'm defining that source in reference to the solute. So water is moving from low concentration to a low concentrated solution. Why is it a low concentrated solution? It has less solute. Then we say it is a low concentrated solution. But the definition shouldn't confuse you. Someone can even define osmosis as movement of water from a low concentrated solution. Why? A low concentrated solution has more water and diffuse solutes. Then someone would wonder, what makes water move? If you've got a beaker of water, like here, let me use the molecules of water with those circles. That's water molecules. This one is water molecules. Now, in this case here, I've not dissolved any solute. That would mean each molecule of water we see here, it has a higher ability to move. Its potential of mobility is high. These water molecules are free to move. Nothing is stopping the water molecules to move. But the moment I dissolve in solute, remember, anything that dissolves in water must be polar. Bipolar, I mean charged, can be negative or positive. And from your chemistry structure of water, water is also polar. I don't know my chemistry, but if I have oxygen here with the two hydrogens, those who have studied chemistry, and all biology students are chemists, unless you are not going to medical school. This oxygen and these hydrogens. Oxygen and hydrogen in water molecule, in a water molecule, which one of the two, oxygen and hydrogen, which one of the two is negatively charged?
Oxygen.
Oxygen is negative charge. And these ones are positive, what? It's charged. Because of negativity, there's polarity. That means water molecules are already charged. That's why they can easily dissolve charged molecules, because there's an attraction, ionic bonding. But I think you ever talked of hydrogen bonding. Now, if I don't go into that, now if I dissolve in here some solids, these are solids which are charged, let me say positively charged. The moment I drop in here, if I dropped in two solute molecules which are positively charged, let me take water. You know what has two charges? Let me take it to be negatively charged. Not forgetting that it also has some positive charge. That means it can take in negative and positive charged ions. But then here, since I'm dropping in positive, let me take the negative charge of water. The moment I drop in this positive charge ions, there will be an attraction between this positive and then the negative, the positive and the negative. That attraction existing between the solute dissolved in water molecule, in a beak of water, will be reducing the mobility of the water molecules. Before I dropped in the solids, all these water molecules, 1, 2, 3, 4, 5, 6, were free to move randomly. But when I drop in the solids, only 2 drops of solids. That means that you check, I had 1, 2, 3, 4, 5, 6, 7. Before solute dropped in the solution, all the 7 molecules were free to move. They had a higher mobility potential. 1, 2, 3, 4, 5, 6, 7. But when I dropped in 2 solutes, some molecules get attached to nearby solutes. If I use this model here, now how many molecules are now attached? 1, 2, 3, 4. The 4 water molecules will get attracted by the solute. That means their mobility will be impaired, will be reduced. Now we are remaining with only 3 with ability to move. You should master that. Why should water molecules move from a low concentrated solution to a high concentrated solution? Is stemming from this. The more solute you dissolve in, the more water molecules will be attracted and stopped from movement. So that means that this solution, this solute, the more I dissolve in solutes, the lower the ability that solution to lose water. Why? The solvent molecules are being attracted. Let me just add in another solute molecule here. If I put in 1 here, it will get attracted here and then attracted there. Now, if I may ask now, if you look at that solution, now how many water molecules in this solution are free to move? Hello?
Teacher, it's 1.
It's only 1. Now, this is what I'm talking about.
Yes.
The more solute added, the more I reduce the mobility of water molecules. Now, if I added another one, you find that you have none. No water molecules here can diffuse. So that means that oh, you're following. Now that means that all molecules here are bound by Okay, what is some technical problem? I brought this man with the special things. Now, you see that, are you still there? Hello?
Yes, teacher. Teacher, we are there.
I think you've seen this principle, the effect of solute dissolved. The solute, its effect, when you dissolve it in solution, it will be stopping free movement of water. And that means that there will be a concentration whereby that solution cannot lose water. Why? It is highly concentrated. All its water molecules are bound by the solute. They are attracted, they can't diffuse out. Now, in this instance here, solution A, this cell, which was inside this solution, which is hypotonic, that means that the cell, it has a higher concentration. It has too much solute dissolved. That means that the water, it doesn't mean that it is dry. No, it has water molecules. But because of higher concentration of solute, or most of its water solutes, molecules, are attracted by the solutes, so they can't move. So what happens? The surrounding solution, where it has a low concentration, that means that most of its water molecules are free to move. Will have a ability to lose water because most of the water molecules are free. So they will diffuse into the cell, which has a higher concentration. That's why it does intervene. Now, when you come to this solution B, where water molecules will be diffusing out, that means that the solution outside the cell, it has a low concentration. So most of its water molecules were free to move. The solution, the cell sap of the cell, it had low concentration. So its water molecules are free to move. But the surrounding is so concentrated. Its water molecules are bound by the cell, so they can't diffuse. So what happens? Water diffuses out of the cell hypotonic solution. But here I want to put in something. There's what you call water potential. Before I go to water potential, at the time which I asked you to tell me about where it can fall. When I talk of osmotic pressure, I'm referring just to a concentration. The higher the concentration, the higher the osmotic pressure. But I want to connect it to here. The higher the concentration, if you have too much solute in a solution, that means that water cannot lose water, that solution, but it can gain. Because its water, most of its water is bound to the solute, is attracted by the solute. So it has a higher chances of gaining than losing. So that means that this solution is hypotonic. It has a lower osmotic pressure. Why? It has little solute dissolved. So it will be having a low osmotic pressure. And this one will have a higher osmotic pressure. But in fact, because here, when I talk of low or high osmotic pressure, think of implication, the effect of that. The higher the osmotic pressure, the lower the ability that solution to lose water, because most of its water molecules are attracted by the sodium. But the higher the ability to receive, to gain. That's the importance of those terminologies. Like here I would say, the cell here has a higher osmotic pressure. That's why it is gaining. And the cell here, which is losing, it has a lower osmotic pressure. But the solution which is losing water must be having a lower osmotic pressure. So most of its water molecules can move. And the one which is gaining has a higher osmotic pressure because most of its water molecules cannot move. Now, there's another term here. There's what called water potential. I think we ever talked about that. Water potential is not talking about the number of water molecules in the solution, but technically it's talking about the number of free water molecules. The number of free water molecules in the solution. Now look at this solution here. Its water potential is very low. Why? It doesn't have any free. If I may put here one water molecule, I would say it has only one water molecule which is free to move. Automatically, it's a bit hard to have all 100% water molecules bound. But then here, I would say almost all cannot move. So when I talk of water potential, I'm talking about the number of free water molecules. So when I say high water potential, it means that that solution has so many free water molecules which can move. When I say the other one has a low water potential, it means it has few water molecules which are free to move. And that would mean a solution with a high water potential, if I relate it to osmotic pressure, I want to ask you that. I'm about to close. Shall begin from there, but let me, if I go. I say water potential, which is symbolized by this symbol, is not measuring how many water molecules are in the solution. Because if I have the same volume of water, I'm not adjusting, and I'm just adding solutes, that means water molecules have remained constant. But the water potential will be varying with the concentration. The more solute added, the lower the water potential. So that's what I'm saying. When you are defining water potential, at the back of your mind, you should know I'm referring to number of free water molecules. And the number of free water molecules vary with how much solute dissolved. The higher the solute dissolved, the lower the free water molecules. And then the lower the water potential. The fewer the solute dissolved, the more there will be free water molecules to move, and then the higher the water potential. Now, I want us to add to place those two, I mean, that term water potential. Between A and B, basing on these, A and B, if you have got the meaning of water potential, which one has a higher water potential? Hello? A and B, which one has a higher?
Teacher, for me, I think it's solution A.
Solution A has yes.
Solution A.
Sorry?
Solution A.
Yes, you are very correct. Thank you. A will be having a higher water potential. Why? It is of low concentration. It has a low osmotic pressure. That means that very few of water will be attracted. Many will be free. So I can say this one has a higher water potential. And this one has a lower water potential. Now, those are the terms that I'm bringing in. But for us in practical, we apply them. We don't define, just apply. Why? We think from your theory, you've known. That's why I've gone through just to rehab the meaning of this before I apply them. But I'm about to summarize. Sorry, I took, before the problem didn't start earlier, but lunchtime. I want to leave you some exercise. You go and read about it so that next time when I come, don't take too much time. In your theory, you talked about this relation. This is in respect to plant cell or plant tissue. We have seen this one. So I want you to go and read about this one. How do you call this one?
Solid potential.
How do you call this one? This is what?
Pressure potential.
Aha. Now, I want you to, during your free time, go and read about this solid potential. It's always negative. I hope you know that. But it's the same as osmotic potential. The other one was osmotic pressure. But this one's osmotic potential. They have the same meaning. Just use different terms to avoid confusion between osmotic potential and osmotic pressure. I want you in your time to find out how does this one vary with the concentration. Of the solution. What I mean here, if I increase the concentration, what happens to solute potential? In other words, between A and B, which one has a lower solute potential or a lower osmotic potential? I want you to find out that. You can read about it. If you have BS, it's all illustrated there. Even the biological, okay, it's biological standards BS, there's also functional approach. And then other texts, go and read about it. How does it vary with the concentration? So that next time, we move smoothly. Thank you for attendance, unless you have a question from what we have talked about before I close.
No, thank you, teacher.
What's the difference between osmotic potential and osmotic pressure?
Osmotic pressure, we have talked about it here. I say the osmotic pressure is just about the concentration. The higher the concentration, the higher the osmotic pressure. And I said the solution with the higher osmotic pressure, that means highly concentrated, that means has higher ability to gain water. and has a low ability to lose. Why does it have low ability to lose? It has a lot of solutes which stop the movement of water because of those bonding. Now the osmotic potential. I don't want to preempt your research because now I'll be answering for you. When you research about osmotic potential, which is the same as solute potential, when you are reading about solute potential, the same thing, you're reading about osmotic potential. I want you to go and find out, then compare with what I have. What is it? How does it vary with the concentration? As you read, as you research, you'll be understanding more than just getting information. We are not just after giving information. You go and research. So that when you come here, we'll have a clear discussion. So I've discussed for you osmotic pressure, but they are different. So you can find out how they relate and how they differ, because the homework I've given. Any other questions? Hope you understand what I'm saying. Should I close? Technical man in Amudala has come. Okay. Well, close till next time. Kati wakazaawo kazaawo ndo abantu bali bafuddeko
anti nkufudde oli wafuddeko nenkufuula host nakyo mwana erinnya biragayo
kati ate awo nyigidde n'embyononamu kati